Okay, so today I'm going to talk about some of our work using DTI and diffusion of kurtosis imaging to look at white matter microstructure across the lifespan. And this is my lab's logo. It's designed by one of our undergrad students, actually. So as you probably know, so white matter microstructure in the brain and spinal cord alike is mostly studied using diffusion tensor imaging. So in the brain specifically, we've been looking mostly at mean diffusivity and fractional anisotropy as measures of microstructural integrity. And they are both based on the assumption of a Gaussian diffusion. So in general, in brain aging, when we observe mean diffusivity increases and fractional anisotropy decreases, which is what both of those are widespread across the brain, then we say this is indication of microstructural degeneration. So just a reminder of the diffusion tensor model. So at a non-zero b-value, we record the signal and we fit it to this exponential model from which we can derive this diffusion tensor. And the diffusion tensor will be characterized by these eigenvalues and eigenvectors, which will then be used in computing mean diffusivity and fractional anisotropy. So here, FA is a function of the second moment of the eigenvalues divided by some form of the mean diffusivity. So for the most part, we, like others in the field, have been observing, as I mentioned, decreased diffusivity and increased anisotropy. Increased diffusivity and decreased anisotropy across the brain. And this pretty much covers the entire white matter skeleton as we show here. But increasingly, large-scale studies, as well as our own studies, have been noticing increases in fractional anisotropy. And this really stands out, as sort of what is the possible cause of this. And in our previous work, we theorized that this is probably because we're imaging in a region of heavily crossing fibers. And imagine this region that I showed before is the crossing of the internal capsule and the corticospinal tract. Well, fortunately, in the spinal cord, there's less of this happening. But if, say, one of these tracts, the secondary tract in this case, degenerates first, then overall, over this entire voxel, we're going to perceive that the fractional anisotropy has actually increased because now we have more of this primary tract remaining. So this was in my PhD student Jordan's 2018 paper. But why do we not always observe this then? Is it because of power? The effect size being small? What could be the reason for that? So we delved into this quite a bit, and I'm just going to show one aspect of this research today. Just as a reminder of the definitions of mean diffusivity and FA, I expanded FA for the purpose of showing that it actually includes an MD term in the denominator, so that while the numerator is the norm of anisotropy, the denominator is diffusivity. So these two variables are actually not independent of each other. And I just put the definition of the second moment of the eigenvalues here. So because MD and FA are not mutually orthogonal, any increases in MD may compete with the increases in anisotropy such that you may see nothing, or you may see the opposite, that FA might be going down. I should mention in the literature regarding brain aging, by far we see very weak or zero fractional anisotropy effect in that region of the internal capsule that I circled before. So I guess this is where we kind of went off to investigate how to get a more unbiased measure of anisotropy. And so this is a very recent paper now published by Jordan in Neurobiology of Aging, in which he discusses some interesting new ideas. So if we look at, again, the definition of anisotropy, if we know that FA is actually biased by the denominator, which contains MD, if we really wanted to disentangle diffusivity and anisotropy, we can just use this norm of anisotropy, which is the numerator here, which is what he proposed as norm of anisotropy NA. In addition, based on an earlier paper which proposed the mode of anisotropy, so this was sort of a slight modification of Hindleman's definition, but the mode is actually a shape measurement. I will illustrate this on the next slide, but these three parameters now together, they are the first, second, and third moments of the tensor eigenvalues. So they form a complete triplet. Now, to illustrate what the mode represents, rule of thumb is that the mode increases if the tensor is more linear. It's a little bit harder to visualize perhaps, but here's an example of how these three variables can covary to create different resulting observed FA values. Remember, FA is the parameter that we're saying is perhaps biased. And if we assume at baseline in this example, this is a cartoon of a young person's diffusion tensor, we have an FA of 0.71, which then is actually based on simulations that use these particular values. So mean diffusivity is 0.8, normal anisotropy is extremely high, 0.99, and the mode is 0.91. So this is a nice and anisotropic and linear tensor. Now, FA can go down in aging for two different reasons, one of which has nothing to do with anisotropy. As our simulations showed, even if NA has no change, just the fact that mean diffusivity goes up as a result of, for example, demyelination, we can expect FA to go down as well. And this you probably expected from the equations I showed earlier. But of course, most often we assume it's the bottom case in which the anisotropy does in fact go down and diffusivity does go up. These are the hallmarks of degeneration. And also the mode of the diffusion is also decreased. In this case, you can see this tensor has become inflated. In this case, we can sort of liken this to when we see dispersion of fiber distributions. So now mode is lower when this tensor is fatter and less streamlined. So these three parameters, does our hypothesis then pan out in reality? What if we apply this model to actual data? Do we expect to see sort of these potential relationships amongst these parameters in real data? And then what is our interpretation? So the paper tests these on a 700 strong cohort from the UK Biobank. These are healthy adults, equal number of men and women divided into 7 equal-sized, equidistant age groups. And the acquisition was a Siemens 3 Tesla system, B-value of 1,000, and the rest are more or less standard. And we used FSR pre-processing and custom MATLAB scripts for the rest of the analysis. So to strip everything away, get to the very basics, these are the maps of the eigenvalue variations with age. I say variations because this is a cross-sectional study, so we're not really saying there are changes, but that is just one of the things our lab is very adamant on. So primary eigenvalue goes up. This basically says the diffusivity is increasing. Secondary and tertiary eigenvalues, they together represent the radial diffusivity. And you can see this discrepancy between the primary and secondary eigenvalues. So in the secondary fiber, as for example, we're not saying this is the secondary fiber, but there is this discrepancy in terms of the different directions of the tensor and how their diffusivity vary with age. So to take this further and to compute our triplet of parameters, Now you can see, whereas we had seen this already before, even if we use fractional anisotropy, we can see this blue across the brain, which represents reductions in in diffusion anisotropy across the brain. But you do see this increase now, this I guess, higher anisotropy in this region that we had seen effects in FA before. So this is saying anisotropy is higher in the older people, as is the mode. So these really do go hand in hand, and they have similar, somewhat similar interpretations. Now, how do we compare? How does NA compare with FA in looking at the same region? I will show that in a little bit, but one other curious thing we found is that in this posterior inferior region, we do not see any diffusivity effect. Is that to say there's no age effect in this region? That is something that I want you to carry in your mind as I go along. So I will try to answer that. But going back to this crossing region, if we were to subtract the effect size of the of age based on FA and based on NA, we find that NA does give us higher effect sizes. So in this very same region, and also extending to parts of the anterior white matter, we actually see NA gives us a stronger age effect. So to quickly summarize that part, so we found that the norm of anisotropy, in terms of its age effects, the effects are stronger than those of FA, likely indicating that when we have crossing fibers, NA is really the proper, the more appropriate measure to use. So NA is a measure of anisotropy that is unbiased by diffusivity. And since FA is a product of NA increase competing with MD increase in that crossing fiber environment, it makes sense that it shows a weaker age effect. And positive anisotropy, normal anisotropy age effects coupled with positive mode age effects indicate selective degeneration at the crossing of fibers. But we also saw in the posterior inferior regions, there seems to be a lack of age effects in mean diffusivity and even in the mode of diffusion. So what does that actually mean? So this is where we bring in diffusional kurtosis. Any questions, or should I just go to the end? So diffusional kurtosis, what does that mean? Kurtosis is a measure of non-Gaussianity of water diffusion along a tract, and it has been taken to reflect tissue complexity and diffusional heterogeneity. So graphically, higher kurtosis produces a distribution of diffusion like the one on top, whereas the lower ones the other way. I would say that biologically, we only expect kurtosis to be greater than 0. If not 0, it would not be less. So this is work that was undertaken by my undergrad student Hiba in collaboration with Jordan. We wanted to compare what DKI shows us with what DTI can show us. So this has been previously done while I was a postdoc, actually. I participated in this project looking at comparing DTI and DKI findings of aging. Of course, at that time, we were not considering this effect of the crossing fibers. So overall, in this paper, which was based on this work in 2015 by Jean-Philippe Coutu, we found generally very strong kurtosis reductions across the lifespan. So as people get older, their kurtosis values are lower, whether it is looking at the mean kurtosis, radial, or axial kurtosis. And in some regions, we might actually characterize that with quadratic instead of linear patterns. But there was not, I guess, a direct quantitative comparison of diffusivity and kurtosis measures in crossing fiber regions, as I mentioned earlier. So as I said, kurtosis reflects cellular microstructure. So if we see a decrease in kurtosis, it generally reflects disruption of microstructure organization. Sorry, there's a typo here, leading to less coherent anisotropy. And so this could happen due to reduced axonal packing density, demyelination, axonal shrinkage, etc. So these are all things that happen in the aging process. And the previous study actually found DKI provided higher regional sensitivity to age than DTI did. But in some cases, in other types of pathologies, we can also see increases in kurtosis, which it may mean the opposite of everything in the above, but it can also arise from increased partial voluming with new tissue types, for example, if there's a tumor growth. So of course, that is not part of normal aging and has yet to be observed. But we do sometimes see this in regions where there is significant atrophy of the tissue, leading to a local increase in partial voluming. But today we'll focus on the fact that we can also get kurtosis corrected diffusivity values, the diffusion tensor that has been accounting for the kurtosis. So this will be different from the diffusion tensor obtained from the DTI model. And so the two types of diffusion tensors also have not been compared in aging. So to do that ourselves, we chose the exact same subjects from Jordan's first study, 700 of them. And we used both the B of 1,000 and 2,000 shells. And we used this Vorat MATLAB DKI toolbox. First, to show the DTI measures, these are kurtosis corrected DTI measures, and they look very similar to the ones we saw before. You still see this general decrease in anisotropy as age progresses, and you do see this mean diffusivity increase, and you do see this increase, this higher fraction anisotropy in the older populations in this internal capsule region. So these are still all here. And more to that, you can still see in the posterior inferior region, there's a lack of MD effects. On the right hand, you see the mean kurtosis effects, and basically everything is going down, everything is blue. So we see while kurtosis does go down in this region, diffusivity does not. To further investigate this, we break everything into their axial and radial constituents. So both diffusivity and kurtosis can be broken down in this way, and we compare them individually. So the first would be axial versus axial diffusivity versus axial kurtosis. And this is sort of a penetration map where you have so purple indicating where we see AK effects, dark green indicates we see AD effects only, and light green, more like yellow, is where they overlap. So just to try to help you read these, what these are showing can be summarized by these 2 points. First, the negative axial kurtosis age effects overlap with positive axial diffusivity age effects in the internal capsule region and the anterior superior tracts. So this is the yellow you see here, quite a large region. But in the posterior inferior tract that we were so interested in, we only see a negative AD age effect, but no effect coming from axial kurtosis. So the kurtosis in this region is not really changing. Not really varying with age. Next is to compare axial kurtosis versus radial kurtosis. So these going in different directions of the axon. So again, this overlap map basically showing the regions in which we see negative age associations in axial and radial kurtosis. And this time the overlap is much larger. So negative radial kurtosis effects overlap with negative axial kurtosis effect in most regions, but not in the internal capsule region, which is purple here, which only has AK age effects, and not in the posterior inferior region, which only has RK age effects, both sort of negative age associations. Furthermore, comparing FA with AK now, again, the same types of overlap, but this time we see that positive age effects in FA overlap with negative age effects in AK in the internal capsule. So now we do see this little region here, which is the only one that is sort of got any kind of overlap in the first row. But if we look at regions in which both FA and AK are reduced in older age, we see that negative age effects in FA are generally without coexisting AK effects in the posterior inferior tracts. So here the light blue shows that FA is becoming lower in older age, but not AK. So here. So radiodiffusivity with this kurtosis counterpart. So here we're I guess the majority of the regions are overlapping in these two senses. So RD is positively associated with age, just as RK is negatively associated with age, except for the internal capsule region, which only sees the RD effect. And the posterior inferior region only sees the RK effect. So these two regions, there's something very peculiar about them. And we can sort of see this recurring pattern. Now, I did mention that we wanted to see what the kurtosis corrected DTI parameters would give us. And here we see reassuringly that most regions exhibit an increase in diffusivity, whether it's axial or radial, except for these posterior inferior regions. Again, here we see axial diffusivity has a negative age association, whereas radial diffusivity has a positive association. So could it be then the reason that we did not see anything in this region in terms of mean diffusivity is that the axial and radial age effects cancel each other out? So there's definitely something going on. It is not nothing. And if we were to look at DKI corrected DTI parameters, compare that to conventional DTI, because sometimes we read papers, we're not sure if the diffusion tensor metrics were corrected for DTI effects or not. And what we generally find is that DKI corrected FA values exhibit weaker age associations, whereas DKI corrected mean diffusivity values show the opposite. And this will be a longer explanation, which I don't have time for today, but I think it's for those that work with these models, it's apparent that in the DTI model, the kurtosis can be attributed to different parameters. So if we don't have a kurtosis term, we will attribute kurtosis variations to either FA or diffusivity. So to quickly wrap that up, we identify these two very odd regions that go against sort of the general trends. The internal capsule region is characterized by positive FA age effect. Also in terms of normal anisotropy and the mode. But in these regions, we also see negative axial kurtosis and radial diffusivity effects, and as well as positive age effects in axial diffusivity. So there seems to be this really distinction between what's happening in terms of diffusivity and kurtosis axially and radially. But if we do not decompose our metrics into these different directions, we really don't know what's going on. One unique thing is that we do not see radial kurtosis age effects in the internal capsule region, which is heavily crossed. So our interpretation of all of these things taken together is that we're looking at selective degeneration of a crossing fiber, the secondary fiber to be exact. And this is consistent with what we had observed before using free water imaging, which I did not show today. Now, turning to the posterior inferior fibers, which are generally lacking any kind of mean diffusivity age effect. So there's a hole there. Seemingly nothing is happening, but that is not quite true. We see this negative radial kurtosis and axial diffusivity effects. We also see positive radial diffusivity age effects, as well as negative FA. So all of these taken together seems to indicate that there's demyelination causing the radiodiffusivity to be higher, but there is also slowed axonal direction diffusion. So AD goes down in that region. And since kurtosis is not changing axially in that region, so the pattern of diffusion is still, Gaussian. It's not any less Gaussian than it was before. So this is probably indicating whatever's impeding that diffusion has no cellular structure. It's probably, debris that are just spread out randomly. So that is the most likely explanation. The interesting thing about that is this happens before demyelination. So the accumulation of debris can happen earlier than demyelination. And if that is shown in our data, it can also be observed in other data sets and can be an earlier indicator of age-related degeneration. So the two takeaways from this supposedly in informal talk, first is that we would encourage the use of an unbiased measure of diffusion anisotropy, especially for studying crossing fiber regions. By that I mean, one simple thing you can use is the normal anisotropy that can be calculated directly from your single shell diffusion data. Does not require any additional acquisitions or even very elaborate modeling. It is very simple to calculate. And secondarily, the added value of measuring diffusion Gaussianity, because it reflects the cellular environment and it tells us why diffusivity might be different, not just that it is different. So two examples are, if we see axial diffusivity reduction without a kurtosis reduction, this might indicate debris accumulation within the axon, which perhaps precedes demyelination. But if we see axial diffusivity going down, but AK also goes down, that could reflect axonal breakdown coupled with, edema, which impedes the water movement. So I think in general, we don't want to do clinically unfeasible acquisitions, but these are quite feasible. I think they're becoming increasingly feasible, and they do give us additional information with relatively little effort. So with that, I wanted to thank my funding agencies and my happy lab, especially Sohiba and Jordan here, whose work I presented today. And here's a photo of Toronto I'll leave you with, although I never see this ever. Thanks, everyone.
Thank you so much, Jean.